Gormination in Sri Lanka. I like to talk to you today about one of my projects, that is the Allelepathic Effect of stratiotes leaf extract on germination and emergence of rice, weedy rice and Saramellagrass under In-Vitro condition. First of all, I like to introduce something about allelopathic activity or allelopathic effect and allochemicals. Allelopathic effect is one of the biological phenomena that the plant produce and release the chemical compound into the environment. It affects the growth and development of neighboring plant. These chemical compounds known as allochemical or allelopathic chemicals. Here I display some allochemical mostly found in plants that are the phenolic acids, phenolics, flavonoids, humanics, terpenoids, and scopodones are the common allochemical present in a plant that can inhibit the growth and development by several ways, such as blocking the cell division, inhibit the photosynthetic process, reducing the germination percentage, like different ways that can affect the growth and development of neighboring plants. The justification of the studies, the finding of the studies can be supporting the sustainable peat management in the agricultural ecosystem, as well as that can support the utilization of invasive plants like Pistia and Iconia like plants are mostly common plant invasive plant present in Sri Lanka. The objective of the studies are: the first objective is the phytochemical screening of Pistia sativa leaf extract, and the second major objective of this study is the investigation of allelopathic effect of Pistia stratiotes leaf extract on germination and the early growth stage of different seeds in Sri Lanka. Here, the methodology part. The first part is the sample collection. The selected plant species is the Pistia stratiotes. This is one of the invasive plant, aquatic invasive plant, widely distributed in Sri Lanka, in east coast, especially in the east coast of Sri Lanka. Heavily polluted by several aquatic ecosystem like the Dicol lagoon. This is one of the selected plant species for the experiment, and selected treatment. The experimental plant seed is the first one is rice. The rice variety is LD365. This is one of the common red rice variety, mostly cultivated in wet and rice zone of Sri Lanka. The reason for selecting this rice variety is compared to other rice varieties, rice varieties must tolerant to the adverse environmental conditions. That's why I select this variety of rice. And the second experimental seed is the weedy rice. Nowadays, you know that the farmers are facing a lot of difficulties to control the weedy rice, because this weedy rice also coming under the same species of rice. So there are no any chemical pesticides, weedicides, so to control this weedicide, the weedy rice. And another common widely distributed grass in the rice zone of Sri Lanka is rugosum seed, commonly known as Saramellagrass. This is also a very harmful weed, widely distributed in rice zone of Sri Lanka. The sample preparation is, first of all, the seeds, the leaf samples were collected from the Manna West area. The Dicol lagoon is present in the area, collected from that area, and allow for the safe drying. And after the complete drying process, the blended into make the fine powder. Then the fine powder, the 10 gram of powder was weighted and mixed with the different solvents such as the water and the ethanol. From that we can get a different extract, filtrates, the aqueous extract and the ethanol extract. Then this extract were concentrated using the rotary operator to get a crude extract. After that we can prepare a different concentration of extract using the dilution of this crude extract using water, the distilled water. You can see the germination test. First of all, the collected 3 different species seeds, that means the rice, weedy rice, and the Saramellagrass were was with distilled water, and after that sterile by 50% sodium hypochlorite to remove the dirt and the microorganism on the surface of the seeds. And uniform equal size of the seeds were collected from the sample. and that are evenly distributed on the varieties that having the wetland filter paper, and treated with the 10 milliliter of different concentration of extract, and also the control treatment treated by the same amount of distilled water. And you can see the treatment setup here. There are 4 different treatments. The control treatment is the first thing. That each treatment setup having 5 duplicates. The first treatment you can see that's 250 ppm. The second treatment 500 ppm and 1000 ppm. And this other treatment explained here. The control treatment is indicated as a T1, and first treatment 250 ppm is T2, 500 ppm is T3, 1000 ppm is T4. And these are the different experimental setup we apply for the different seeds. For example, the aqueous extract of stratiotes apply the rice wheat. At the same time, the aeronautic extract of stratiotes apply the different kind of wheats as well. These are the parameters we observe after the 7 days period. The first thing is germination percentage, premium and radical length. These common parameters we evaluate for the evaluation of allopathic effect of different extracts. Okay, these are some photographs, the statistical analysis done by the SPSS version 2020. And these are the results from the phytochemical study. We got that the allochemicals present in the plants is the aqueous extract having tannins and phenolics. At the same time, the aeronautic extract having tannins, flavonoids, terpenoids and steroids. From that, the phenolics and flavonoids are having more allopathic potential. Here, the first result is related to the allopathic potential is the germination percentage. You can see this graph. Directly we can get both the aqueous extract and the aeronautic extract showing the significant inhibition of the different seeds. Here, the rice seeds showing slightly increase the germination percentage as aqueous extract, but other the aeronautic extract that's showing slightly reduce the germination percentage. But the other weeds, the wheat rice and the Saramellagrass, that's showing the reduction of germination percentage with increasing concentration. From that we can get some idea from that the Saramellagrass is more sensitive to this kind of different extract. For example, we can see this both graph, the control treatment that's showing the significant amount of germination percentage, but the different extracts showing the notable reduction in the germination percentage. Almost none of the seeds are germinated from the aqueous extract in the each concentration. And you can see these pictures. At the same time, this radical length and the premium length also measured. This also inhibited by increasing concentration. It is control treatment having so more radical length and the premium length. At the same time, the aeronautic extract and the aqueous extract showing very less premium and radical. From the results, we can conclude that compared to both extract, the aeronautic extract showing more allopathic potential in the aqueous section, and also compared to the other seeds, the Saramellagrass seeds showing very sensitive to the other both extracts. From that, we can develop this research and separate these chemical compounds from the leaf and help for the development of eco-friendly and alternative weed management in the agriculture system. So, reference and